Welcome back to this second segment, if you will, in the series of 3 ABC presentations on the nervous system. I have retitled this segment the -2 to emphasize what we'll be covering. It will be membrane potentials, graded potentials, and action potentials. And then, of course, the action potential conduction down the axon. We already considered the myelination. So the nodes of Ranvier will relate it to that. And then throughout, of course, we'll be reflecting on the role of ions, and we'll end with a discussion of the impact of changing levels of plasma potassium levels, potassium ion concentrations, electrolyte balance, imbalance, and how it might impact some of the parameters that we're considering. So the first quantitative assessment of membrane potential that we want to consider is in the context of referring you back to the introductory chapters where resting membrane potential is considered, and this equation, the Nernst equation, is first introduced. We will essentially review and repeat much of that at this time. But still, you should go back and review that information to have a better appreciation of how resting membrane potential for any cell type is generated. To let the cat out of the bag, it basically is related to the concentration of ions, charged molecules. The Z is the charge. And how many charges? Like potassium has 1+ charge, right? And the concentration difference across the membrane. We'll get into this in a little more detail as we go through. And then if you have it moving from one location to another, in, out, or out to inside the cell, then it's going to generate a current because you have a charge that's moving, and it can alter the potential difference across the membrane. So this is the basic idea or concept that we want to have in mind, and now let's just try to picture what we would need to see in a cell for us to be able to put a recording electrode inside and a reference electrode outside and measure the membrane potential difference, or the potential difference across the membrane. So it's always with reference to the outside. So is it more positive inside than outside, or more negative outside than inside, which is saying the same thing? But that's what we want to be thinking about at this time as we introduce this formula. It takes into account not just the concentration and the charge, but other factors represented by this 61, such as temperature and some other constants, as we'll see. And this gives us a value, the Nernst potential, for that particular ion, which is in the subscript. So it would be a K with a plus for the equilibrium potential is another term for it, the Nernst potential, Nernst equilibrium potential, to put it all together, for potassium. So this describes the membrane potential that a single ion would produce if the membrane were permeable to only that ion. So we don't take into account the degree of permeability to the ion in this formula because we are considering it to be absolutely permeable, if you will, right? So what would happen if you could make a membrane completely permeable to one ion only, and you tracked the membrane potential? You say you started at one point and then you let it go, and you could track it and see the change over time, and then you reach the end point. What would it be at the end point? That's what this formula will predict or calculate for you. Now, if you follow the arrow here, I'm actually going to change it to a felt tip pen because I want to make that point. In physiology, it's very important to understand this continuum, that there is a start point. You have an initial, I'll put a little I in there, That's kind of a way that we have, whoops, I didn't get to dot my I. There we go, put it up here. We have our initial state, and then there's going to be some period, time interval, over which there is going to be a dynamic change of whatever parameters we are observing from the initial state to the end point. And I'll just put a little e in here for the end. And then we're going to have, I'll put S1 here and S2 here if we're looking at initial state and end state, and then some series of S sub d's, if you will, the dynamic states throughout the dynamic process. So whenever you're studying physiology, physics is the same way. You need to know where along this process you are making the assessment to understand what your answer would be. So an end state is what we are predicting with this formula. If you started at some condition, an initial state of however much potassium is inside and however much is outside your artificial cell or in vivo cell prep where you can control these things, and then you open the potassium gates and you allow it to proceed to the point to where the concentration gradient pushing the potassium out of the cell is balanced by the electrical gradient trying to hold the potassium inside the cell, or according to the textbook, pull it back in, but it's really holding it within, resisting the outflow. So you've got an equilibrium of potassium in and out. At this end stage, what would be the membrane potential difference inside versus outside? So that's what this formula is telling us. For only one, get to the arrow, and then I got to set it back to be visible. Thank you for waiting for that. For only one ion, and when we are not concerned about the degree of permeability, we're considering it to be a complete or absolute permeability. So this formula works perfect for that. But knowing what the value would be for this, the equilibrium potential for any one particular ion, later we're going to have the benefit of being able to predict the direction of change from state 1 to state 2 for a known biological system when one or more ion channels change in their permeability, either becoming more permeable or less permeable. So if we have a complete biological system, as we proceed through this, we'll see it and state it again. If you open the potassium channels, then the membrane potential is going to change in a direction toward the equilibrium potential for potassium. So let's proceed through this series of slides to get a better feel for that. So hopefully you have a feel for the practical utility of the Nernst equation, because we'll go to another one that's more commonly used, but still this is the key one to be thinking about what I just said. When you start from a biological state, a resting state, and you change the permeability for one or more ions, then and you know what the membrane potential is at that state 1, what will you predict it might be, or what would be the direction of change in the dynamic or at the end point after that event? And then we'll take it into account when we talk about the action potential. Different phases of the action potential, actually. So the membrane potential is influenced by the concentration gradient of the ions across the membrane and the membrane permeability to those ions. This condition is considering, as we said, the complete permeability. Well, I always call this the Goldman equation, and forgive me, I am not recalling the H and the K, do you? Goldman, Hodgkin, and Katz. Yes, I just turned to the page in the textbook, as I would expect you to do, to look that up. So the GHK, for short, equation is shown here. I always refer to it as the Goldman equation, forgive me. Excuse me for clearing my throat there. This equation predicts the membrane potential that results from the contribution of all ions that can cross the membrane. As you will note in the text, calcium should be included in this consideration, but rarely does calcium contribute to the membrane potential. So its permeability typically, even though the concentration gradient might be quite robust, the permeability is negligible. So this p-value here is the permeability for that particular subscripted ion. So this is the permeability for the potassium, and you see that it is present in both the numerator and the denominator. But for the P sub Ca, calcium, is typically around 0, or so close that you can just eliminate it from the equation. So you see it excluded from the version of the Goldman equation, or the GHK equation shown here. So this one lets us think of the physical properties, the biophysical properties of a living cell more completely, because we know that permeability can change over time. So this value of permeability will not always be exactly the same from one time point to another time point for any one particular cell, unless it's just sitting there like a somatic cell that doesn't depend upon changes in its membrane potential. In other words, it's not an excitable membrane, but it does have a resting membrane potential because the concentration gradients are going to exist. But if the permeabilities never change, then there's no major change in the membrane potential. So the V sub n is the membrane potential in voltage, and we'll see the common unit is millivolts. So if this was V sub m res for resting membrane potential, what would the value of this be? And that at any time interval subsequent to a resting membrane potential for an excitable membrane where permeabilities are going to change, then what would be the membrane potential at that instant in time? So again, this is the way to begin thinking about this. We're talking about instants in time, if my drawing is still there, instants in time across this dynamic range of events of changing from state 1 to state 2, where we might be taking snapshot pictures of things and then either measuring the dynamic change between the two states or trying to imagine, picture, or predict what would be occurring. And the permeability is what's changing. So another key important aspect about this is that in biological conditions across a membrane, you are going to have current, the flow of potassium or sodium or chloride, and you're going to have changes in permeability, but the sodium concentration outside the cell and inside the cell is not going to change. You'll have a very localized, just across the membrane, difference. And I want to illustrate this here. This is so important that we need to think about it. So if we illustrate the membrane here, and this is out, and this is in, we always orient to that, right? And so sodiums are all over, right? So sodium I'm going to show is something like that. So they're all over the place, right? They're everywhere out here. And there's more out than there are in. And then there's K's, potassiums, and there's a lot more potassiums inside than outside. And some of them are far away from the membrane, and some of them are close to the membrane. Same thing for the sodium. And then there are chlorides, more of them out than in. And you might find those that in more frequently are going to be right next to the membrane, not just all in the same spot. I'm making the mistake of putting them only in one location, but they're randomly, evenly distributed throughout, right? There shouldn't be any force that's saying, you sodium, you stay right there. To make them move, that's the current flow that we're going to be considering. Yes, there are forces that can cause sodiums to move along the surface of a membrane or to cross in or out to generate a flux across the membrane, but we can also have a flux along the membrane. Think of being in a crowd where you're a bunch of sodium people all lined up in the street, and somebody gives that one person a push, then everybody's going to kind of shift that way. Or a wave in a stadium is the one that we'll think about when we talk about a current moving along a membrane. So you can have sodiums move. So remember, if you've got a recording electrode here, and you're measuring a current, and a current is the move of a charge with reference to a point location at an instant in time, then if you get a sodium that goes across there, even if it comes back, then this sees that as a movement of charge, and that's a current, that's a flow of a charged particle, right? So you need to think about what really is being measured when we go through all these conversations. So we may be changing so that there's more sodium coming inside the membrane, all lining up close to the inner surface of the membrane. And then if you've got pluses here, what happens to the negatively charged molecules scattered around out here? They get pulled in. So you now have negatives close to the membrane there. But did you have so much sodium move into the cell that you now have a higher sodium concentration inside than outside? No. But a very localized event here, so there's more sodium here than there is on the outside, so you get a plus on the inside and a minus on the outside. And normally, of course, it's the other way around, with the sodiums close on the outside, and the negatively charged, not so much chlorides, but there's proteins inside cells, anionic proteins that have a negative charge on them, and they're the negative guys, as you know, that would be contributing to that negative on the inside. So this is the way to think about it, just at the membrane. Don't think the whole cell. So you're not going to change K in and K out, but you're going to change the permeability value here, and nothing else is going to change. So to change the membrane potential, all you need to do is change the permeability. And this concentration difference inside and out is going to be robust. So you change the permeability, and it's going to have an impact, a big impact on the observed dependent variable, if you will. Okay, go back to arrow, and so I'll try not to switch anymore, but I hope these illustrations are helping you to envision what we're going to be talking about collectively, and I try to put it in the perspective of these formula because they can seem kind of daunting at first. It's like, oh my God, it's the log and all this other stuff. And what it comes down to is we just have to think about changes in permeability. So what are the physiological properties of membranes that regulate selective ion permeability? So your review lectures should help you to recall some of this information, and obviously it's ion channels or membrane proteins that are going to be related to that. So the resting membrane potential is determined primarily by the potassium concentration gradient. So that's not going to change unless there's something that changed it, and that's what we'll discuss at the end of this presentation. So under normal conditions, you're not going to change the interstitial fluid concentration of potassium or the intracellular potassium concentrations. So that's going to be ever present, and it will be a constant. It can change, but if homeostasis works, it should be a constant value. So the permeability is the critical thing. So at rest, there are what are called leaky potassium channels, ion channels in the membrane. So potassium is constantly leaking down its electrochemical gradient, more so the chemical gradient, trying to escape from inside to outside. And then, of course, you know that there is the sodium-potassium ATPase that is taking whatever sodium that is leaked in, 3 of them, and pumping it out at the expense of ATP and bringing in 2 potassiums every time it does that. So it's a sodium-potassium anti-porter that is ATP dependent. The sodium-potassium ATPase. Everybody should have heard about that by this point in your education. Okay, so this is the counteracting effect for the leaky potassium and leaky sodium characteristics of the resting permeability of the cell's membranes. And of course, chloride is leaking a little bit, but the biggest component, the major player of these 3 for resting membrane permeability is the potassium. So the gated channels control the ion permeability. Excuse me. So here what we are talking about is state 1, the resting membrane potential. And then to alter state 1 from resting membrane potential to any other state, or the dynamic process to achieve that second state, we are considering gated channels. And the gated channels are the ones that are controlling the ion permeability, the p-values in the Goldman-Hodgkin-Katz equation, right? So, they can be mechanically gated, such as a pressure sensor receptor, a stretch receptor in a blood vessel, the aorta, or the carotid sinus, or the intestine, or even in your skin, right? Or even in the terminals in the muscle, or even going on forever. Or chemical gated, such as ligand gated from a peptide, a water-soluble molecule. So we've talked about ligand-gated ion channels in the first series of lectures, so there's no surprises for you there. And the voltage-gated, we also saw those. We at least mentioned them, and we'll look at them in more detail in this present talk. So the threshold voltage varies from one channel type to another. So we're going to talk about, obviously, potassium and sodium and chloride channels. And there are voltage-gated potassium and sodium channels and voltage-gated calcium channels that aren't in the context of this discussion. And typically, we're going to see chemically gated chloride channels. So, what are the properties? What determines when and how a potassium channel will open, or the rate of opening, and when and how it will close? Same for sodium. So is it a mechanical event to open, a timed event to close? Is it chemical dependent, voltage gated, or dependent to open and close? So these are some of the characteristics we're going to consider as we talk about these. So each will have a threshold for opening and maybe a different rate of opening, and in some cases that rate of opening or extent of opening or the threshold can be modulated. Remember that talk about how the metabotropic receptors can, through their G coupling, affect a nearby membrane ion channel. We could affect the threshold voltage for it, the rate of opening, the extent of opening, and therefore how much of ion would pass through over any period of time, i.e., that is, the permeability change over time that we would be affecting. So this is where that prior information is coming to play here. And so I'm hoping that in your mind you're seeing that image from the previous presentations of how the metabotropic receptors that are ligand dependent could impact a nearby voltage dependent ion channel in the membrane. Now then, let's take some time to look at this table, the comparison of graded potential and action potential. So we have different features of cell membranes, and here we can just talk about neuronal membranes because this is the nervous system we're considering specifically. So anywhere along the surface of a neuron and the anatomy was just reviewed in the previous presentation. So there's just picture all the dendritic surface, all the soma surface, the axon hillock, the full length of the axon, and the axon terminals. What kind of membrane proteins are you going to see in those areas? And now we're specifically thinking about sodium, chloride, calcium, and potassium ion channels, and the sodium-potassium ATPase pump, another membrane protein, scattered throughout that entire membrane surface area, I'll use that term, and where will you find each of them at the highest concentration, at the lowest concentration? Right? So the voltage-gated calcium channels we're going to find mostly at the axon terminals associated with the exocytotic event. So the highest concentration of the voltage-gated sodium channels is going to be at the spike zone, the trigger zone, and throughout the axon. But the highest concentration at the trigger zone, where you're going to make the decision or set the threshold for voltage for triggering an action potential. So this anatomical distribution of these membrane proteins, and somehow the neurons deciding which voltage channels, which proteins to put where and at what concentration, becomes much more sophisticated as we think of it this way. And it's very intriguing now to realize that neurons are making these important decisions, in quotation, of how they are going to be, their membranes are going to be structured, and therefore determine their sensitivity to input, their integrative nature, and their ability to generate a certain type of codified signal. So very sophisticated. But anyway, this is the reason for bringing that up at this point, because obviously on the dendrites and the cell body, the membrane is not going to have the highest concentration of voltage-gated sodium and potassium channels. It will have other types of ion channels, and therefore you cannot get an action potential, the all or none, but you can get a graded potential. So this is an analog signal. It is graded to any level, 0 to whatever other value there may be, right? So that is an analog signal. So the neuron is an analog to digital converter. But remember, if we're thinking about a synaptic event generating the graded potentials, then we have the chemical signal at the synapse, moving to the left here, picturing a synapse contributing to the formation of a graded potential. That's the analog that was produced by the digital signal, the action potentials in the axon, the presynaptic axon before this. So try to keep this integrated activity going on in your mind. Don't just think about a graded potential. Say, where is it? What brought it about? What happened before it? What caused it? And what does it cause? What does it produce? So now let's just go to the table as we hopefully have a better feel for graded potential, and we'll look at them in more detail, obviously, and an action potential. So we have analog to digital conversion going on, and that's what we're comparing. So how does the analog membrane electrical signal, or change in membrane potential, differ from the coded membrane potential event, the action potential? So this is the input signal. So it is the analog signal taking into account all the input to a neuron. We still haven't yet seen the anatomy of the total synaptic input to a neuron, up to 10,000 or 20,000 on a single cell. So a lot of integration of analog signal coming in. And then the action potential is a regenerating conduction signal. Once you generate the digital signal, it regenerates itself in order to conduct down the axon. And we'll see why it can't go backwards. Where does it occur? So what's the anatomical localization? We said the dendrites and cell body, and then obviously the trigger zone is where we want to initiate the action potential, and then we want to have a mechanism by which we make sure that it only goes down the axon away from the cell body. So the axon carries the signal away from the cell body. And this is where we'll have one slide where we want to remind ourselves there's a difference between the normally functioning cell or somebody putting, and we use this clinically as well, the TENS, transepidermal neural stimulation, TENS. So we either stimulate the muscle directly or put it over a nerve to stimulate the nerve. Well, in that case, the action potential goes wherever it can. So we'll look at the molecular and biophysical properties of the conducting zone of the neuron, the axon, and the trigger zone together to see how unidirectional conduction is regulated normally and how it can be exploited experimentally or clinically. So there are types of gated ion channels involved, so mechanically, chemically, or voltage gated for the graded, mechanical for our sensory receptors predominantly, but some chemical, like taste and olfaction. Then the voltage gated channels, but chemically we typically think of neurotransmitter or neurohormone. The voltage-gated channels we've talked about, and we'll see much more about that. And then the ions involved, typically sodium chloride and calcium, are going to be involved in generating and integrating these signals for the graded potential. And then the voltage-gated sodium and voltage-gated potassium, as we said, are really going to be important for the action potential. So you have more players, a more diverse set of players involved in the graded potential for the analog signaling, whereas the digital signal, you just have two players, and they're countering each other. This will depolarize the membrane, this will repolarize the membrane as far as producing a square wave pulse like a digital signal, right? Sodium goes up, you're on, sodium comes in, membrane potential increases, then that sodium channel closes, potassium opens finally, and potassium escapes the cell, and the membrane polarity comes back. So in effect, you've got an action potential, a square wave pulse event. I'm not saying that the action potential looks like a square. You know it doesn't. But to kind of convert it to electrical engineering type of terminology, it would be the same as a square wave pulse. So the type of signal then is a depolarizing. For example, if sodium is moving into the cell across the membrane, or hyperpolarizing if you get a negative charge coming in like the chloride. The action potential is always depolarizing from resting membrane potential. Remember, always ask, well, what are we talking about? Are you talking about the dynamic, state 1 or state 2? So this action potential, they're talking about the process of going from state 1, resting membrane potential, all the way to and including state 2. So it's a depolarizing event, depolarizing signal. Even though there's repolarizing to shut it off, the real signal is the depolarizing event. So the strength of the signal depends on the initial stimulus, and it can be summed. So it's an analog signal. They can be additive or subtractive or potentiating, as you might know. Then the action potential is a digital signal, so it's all or none. So in this sense, we consider it a square wave event, right? So it's a smooth wave, as you know, because it's a biophysical property of the membrane. But as far as codified signal is concerned, it's either on, all the way, or off. And it cannot be summed, so you can't push two action potential events together on the membrane and have a greater magnitude. So remember, the action potential, we had talked about this a few times before, and we'll continue to remind ourselves, it is not AM type of a signal, amplitude modulation. It's FM type of a signal, frequency modulation. But we have one other component than just frequency modulation. We have the interpulse interval. So you can send a 5 hertz pulse and then how long before you send the next pulse, and will it still be 5 hertz? So that interpulse interval is another feature of the codified signal, but you can't sum them in any way. You can change either of those two, the frequency modulation, but you can't sum or subtract them. We'll see how you can cancel it out through a type of axoaxonic inhibition, but let's just not worry about that at this time. So what initiates the signal? So the entry of ions through channels, and then of course there's going to be some threshold for the voltage-gated event. So the graded potential at the trigger zone is going to have to reach a certain point to impact the sufficient amount of the population of voltage-gated sodium channels at their highest concentration in the trigger zone. So again, I get this concern about these highly reduced illustrations that are showing like 1 sodium and 1 sodium channel. Think of the trigger zone as a cone-shaped membrane structure, the end of the neuron and the beginning of the axon, and you have a high density, but a population of voltage-gated sodium channels. And you must ask yourself, at any one point in time, what is the state of each and every one of those? And within the population of sodium channels in this trigger zone, are enough of them available to be opened, and now have I opened enough of them such that they can trigger a self-regenerating signal that can be conducted down the cable, right? The axon. So remember, our neurons are going all the time. So inside our brain right now, if you think you're going to find a trigger zone where every single voltage-gated sodium channel of the entire population of that cone-shaped axon hillock is in resting state and ready to go, then wake up, start listening to the lecture, and get your neurons going again. It's just not happening. There's a certain set of that population that are in this state or that state all through the dynamic process of being activated, opened, and the inactivation and closing and resetting process. And we're going to look at the anatomy, molecular anatomy of that, as you might anticipate. But this is the time where we're considering the difference between this analog signaling and this digital signaling, of what it really means. You have to have a certain density of voltage-gated sodium channels present in an area and available to be opened to trigger an action potential that can regenerate itself and conduct away from that site of origin. The molecular regulation of the biophysical regulation of the ion channels for graded potentials is going to be a little more subtle because we have an analog signal, right? But there are some voltage-gated sodium channels distributed throughout as well as voltage-gated calcium channels distributed throughout the soma and the dendrite membrane. And the textbook even mentions that the scientists now accept that you can have an action potential as kind of a retrograde signal backed into the cell to let it know, oh, you just fired an action potential. So take that into account as you are interpreting and integrating the entire analog information data set that you're receiving, right? So integration is analog, but once the decision is made, you get an event at the trigger zone that reaches threshold. You have very few choices. Fire an action potential? Yes. Okay, if you're going to fire action potential, how many? And when are you going to stop? How long are you going to wait before you start that series again? So that's the series of decisions made at the trigger zone, but dictated by the integration of the analog signals. So the strength of the signal depends upon the initial stimulus for the graded potential, and it can be summed, so you can integrate. So the initiation is the entry of ions through the channel and its threshold for the action potential. So let's then settle on the description here of the unique characteristics. There's no minimum level required to initiate, but there is the threshold required for the action potential. Right? And it is related to the population of voltage-gated sodium channels available to be activated at the trigger zone. And subsequently anywhere along the length of the axon, of course. So two signals coming close together in time will sum, or in distance, as you know. And then the refractory period is going to be a very important feature of the action potential. So two signals too close together cannot sum. So in other words, we have to think about the state of the population of sodium and potassium, the voltage-gated sodium and potassium ion channels. Is the membrane ready to fire another action potential? If it isn't, it's refractory, and you can't do anything about it. You have to simply wait until it's ready again. So the initial stimulus strength is indicated by the frequency of a series of action potentials. So here then we are seeing that idea I had before. Think about what came before the graded potential. It is a stimulus, a codified signal in the presynaptic neuron. It was the action potentials, frequency and interpulse interval, and therefore a number of synaptic vesicles dumping a certain amount of transmitter into the synapse. So that's the initial stimulus strength that was determined by the frequency of a series of action potentials. In other words, sets at a certain frequency separated by a certain period of time, the interpulse interval. And that's all we have to work with in the nervous system. So here's the presynaptic portion that we just talked about. You have frequency and interpulse interval coming over a period of time. So it might be active at a hertz of 10 with an interpulse interval of 100 milliseconds for a period of 80 seconds, whatever. Maybe that's a long period of time. Might even just be 800 milliseconds or something. So you only got 8 sets of pulses, and then that releases the transmitter to a certain degree, and then that is going to have that degree of effect on the postsynaptic ligand-dependent ion channels. In this case, it's showing a sodium one, and so a certain amount of sodium is going to be introduced into the underside of the membrane near the synapse, and then that creates a local depolarization, and the synaptic event occurs. So we should be talking about that in later lectures, whether it's a neuron or a skeletal muscle. And then that is going to have a spreading effect in every direction. And so closest to the synapse is going to be the largest magnitude, and as you cover a distance, you see it's shown 1 through 5 in every different direction, such as B or A. Think of this three-dimensionally. If you were floating in space up here looking down, you would see this red spreading every which direction that it could, as long as there's no barrier to stop it. And it also, if you were standing above this area looking down, you would see a cone-shaped red gradation. Very red at the central part, the point of the stimulus origin, and then that red intensity, or the magnitude of the change in membrane potential, or its amplitude, the strength of the graded potential in millivolts, shown in the x-axis here, would be decremental. So the farther away from the origin, think of it as sounds. Somebody stands in one location and shouts, and the people closest by are going to hear the sound at the highest magnitude, and it's going to degrade to the point where you can no longer hear it or experience it at some distance. So that makes it graded. That over time and over distance, the magnitude will not stay the same. It is not a continuous magnitude over distance and time, like an action potential would be. So in this case, there's no amplification, so there is going to be a deterioration. So the signal degrades over distance and time. Okay? So the graded potentials decrease in strength as they spread from the point of origin. So the signal intensity, the amplitude, degrades over distance and time. Two ways of saying the same thing. So what we have to picture, and I wish they had shown the picture here very soon, is just how many synapses are we talking about? This is one, and so on the whole surface of this cell there would be 10,000 of them. So how many are doing this at what point in time? I guess down here is where we're looking at the synaptic input. So 2 out of 10,000, are they going to have any effect? How can 2 out of 10,000 have any effect on the integration of this, the synaptic innervation, the global synaptic innervation of this neuron? Well, depends upon the coded signal coming in by any one of them. One of them could cause that neuron to fire a burst for a period of a few hundred milliseconds. So it depends on many different things. One is location. If it's an axosomatic synapse, especially if it was right near the axon hillock, it might have a bigger effect to excite the cell membrane nearby and reach threshold than one that is way out. I mean, some of these dendrites go out 100 micrometers or more. So one way out there might not have as much of an effect as one right close to the, so it's very sophisticated when we start thinking about the real life anatomy and organization of the synaptic input. But it comes back to these same basic premises that we see here. It depends upon the stimulus of the presynaptic input and the properties of the postsynaptic membrane and the adjacent cytoplasmic membrane, whether it's soma or dendritic. What kind of proteins are there? What are the channels that are there? What's their density? Do you have enough voltage-gated sodium channels here to help sustain the higher amplitude over a greater distance and time? But it really depends upon the codified stimulus here, the digital to analog, the concentration of transmitter, how many receptors are affected, and therefore how much sodium or change in membrane potential occurs originally at this site. And so you get different magnitudes here depending upon those parameters, and that initial magnitude will impact time and distance effects. So in other words, if you start with a larger graded potential at the site, it's going to go a greater distance and persist for a longer period of time, because the rate of decay, depending upon an individual neuron's membrane properties between that synapse and the axon hillock, the rate of decay over distance and time is going to be pretty constant unless the neuron decides it needs to change that in pathology or with physical demand, such as when you're lazy and hanging out on the couch versus working out every day, right? Or taking the summer off and not learning, and now you're learning, your neurons are going to respond and start putting better membrane proteins in here. What a way to say it. But anyway, strengthening the likelihood that you're going to be able to learn from listening to me talk so much, getting it to the right places to form enough action potentials to convert this into a long-term memory, right? So neurons don't just sit around and do the same thing all the time. They change based upon what we ask them to do. They have that flexible plastic capability as far as differential gene expression would be concerned. Okay, so let's talk about subthreshold and superthreshold graded potentials in that context then. So not every single neuron is exactly the same, and from day to day, year to year, they are not exactly the same. But the same basic biophysical properties of distance and decremental decay over distance and time do apply. Okay, so here we have a certain stimulus. You're concentrating hard and sending a certain frequency in an interpulse interval, generating a certain synaptic concentration, a certain amount of sodium coming in here, and we measure the event very local to the synapse. So it's not the synaptic event, it's the nearby membrane, the graded potential that is spread from the synaptic event. And we see from resting membrane potential, state 1, over time, the dynamic event, we could go here to state 2, saying state 2 we chose to be the peak membrane potential change over this full event back to resting membrane potential. And we see that at that state 2 that we selected to look at it, we were able to measure a minus 40 millivolt membrane potential that at that location at a different period of time went back to resting membrane potential. So we did get a smooth wave event that gave a total deflection from minus 70 all the way to about minus 40, closest to the stimulus. At some distance away, roughly a little bit more than halfway to the trigger zone, I call it trigger zone because we're thinking function, we record there, and what is the deflection that we see? Well, relatively same resting membrane potential because the ion channels and the leaky potassium, leaky sodium, and the sodium-potassium ATPase protein membrane concentrations are relatively the same here as they are there. So we see resting membrane potential essentially the same, but the change that is experienced is less. Why? Because some current will leak out of the membrane just passively as it is migrating across the membrane. So current can be lost across a cell and just dissipates, and you get a decrement in the total size of the transmembrane differential, right? So if you leak current out of the membrane and it just disappears across this distance, then the change in membrane potential, distribution of charges across the membrane, inside versus out, is going to be lower as you get farther away. And then when you get right close to the axon hillock, same resting membrane potential, you just don't have enough juice left, so to speak, to reach threshold to trigger an action potential. So this is where I said that your distance away can matter. So if you want to be a very influential input, and of course, dynamically through our in utero development and then development throughout life, these things are set up and established and maintained. The most influential ones could be right here to say, well, when I excite the membrane here, just a little bit of excitement is going to get there and be super threshold. But if we consider the same distance away, then we have to concentrate harder to form a memory if we want to really extrapolate it to something. If you concentrate at sub-threshold while you're paying attention to this presentation or reading the textbook, it just doesn't get through, right? So it's sub-threshold here. It doesn't produce enough excitation of enough neurons to form a memory. And we're reducing a whole population of neurons in our memory circuit to this one little blue cell here, but again, what happens at one cell is happening on the global scale, so keep that in mind. So if you concentrate really hard, a large enough population of neurons are going to experience the increased stimulus input so that the graded potentials are going to be still at threshold when they reach the spike zone, such that you're going to have action potentials, and it's going to activate your memory circuit and create all these associations going, I remember this picture, I remember this being higher, this one exceeds 40 here, so when we go to that state 2 that we looked in the previous one, state 1 resting, state 2 at the peak deflection of the membrane potential, closest to the site of stimulus, up to minus 40. Well, now we exceeded that up to like minus 45, I guess, right? And so that little extra boost to begin with, that little extra focused concentration, means that we're closer to 40 at the second location, halfway to the spike zone, and we exceed threshold by the time we get there. So notice that on the time scale on the x-axis, the time of stimulus is here. So the point at which you measure the graded potential at 1, 2, and 3 is different. The shortest period of time you measure the first one, the intermediate period of time you measure the second one, and the longest period of time subsequent to stimulus you read the third event. So there is the spatiotemporal representation of the stimulus and the response on this graph. And of course the y-axis is the typical membrane potential in millivolts, in units of millivolts, and typically you will see it from minus 70 for the graded potentials, maybe to a minus 20 or 30 or just to 0. For the action potential, we're going to have to go above 0 on this y-axis to get into the positive range. Why? Because here we are dealing mostly with potassium contributing to resting membrane potential, and just a little bit of sodium is coming in. And so at resting membrane potential, what ion is contributing predominantly to the current? Potassium, the leaky potassium. So if we think of the Nernst equation, then the Nernst, the equilibrium potential for, and I wish they had showed that table in here, but it's in the textbook. The equilibrium potential for potassium is minus 90 millivolts. So we're not at minus 90 here at resting membrane potential, we're at minus 70, but still, because we're so close to the equilibrium potential for potassium, we figure potassium's the major ion that's moving to generate this. So because it's mostly potassium here, we can think, well, it should be close to minus 90, and it is. And then at this event, it's mostly sodium that's moving, so then we should predict that the membrane potential is going to change. Let's go back here where it shows the sodium coming in. So the membrane potential then at stimulus is going to be deflected toward the equilibrium potential of sodium. The equilibrium potential of sodium is plus 60 millivolts. So we're going in that direction because it's mostly sodium contributing, but not enough sodium goes through to reach the equilibrium potential of sodium. Well, obviously, if we are at threshold and we generate an action potential, we will get closer to that. We'll bypass 0 on the y-axis and get closer to the plus 60 equilibrium potential of the sodium. Now the chloride is minus 63 millivolts for an equilibrium potential. So it's also contributing to some of this analog integrated signal. And when we look at the inhibitory influences to blunt these effects and minimize these, we're going to be taking into account its minus 63 millivolt equilibrium potential. So this is where the Nernst equation, the Nernst potential or Nernst equilibrium potential comes into play for us to ask questions about what's happening at this point in time to generate this membrane potential. What's happening at this point in time to generate this membrane potential? Because it's at minus 70, it's got to be either of two ions, the major players, either potassium or chloride. And we just know that for resting membrane potential, it's mostly the leaky potassium that's going on because chloride's not the chloride permeability at any one point in time in the resting membrane is not really great, but for potassium it is. But we can have ligand-gated chloride channels, so that's what we're proceeding to.